So, good afternoon, students. That's the second question of seminar 3, the mechanism of the receptors, excitation, the receptor and generation potential, and the adaptation of receptors. So first, let's revise what is the receptor potential. So receptor potential is a kind of potential that of membrane potential, but the difference between receptor potential and action potential is receptor potential is graded potential. What does it mean it's graded? So if you remember in action potential, it was following the rule of all or none. What does it mean? So it means that either the action potential happens fully or it doesn't happen. So when the stimuli exceed the threshold, action potential will happen fully with full strength and that's it. Or it doesn't happen if the stimuli was below threshold. But receptor potential, it's graded. What does graded mean? It means that the stronger the stimuli, the higher the potential. It means it's changing with the intensity of stimuli. So as you can see, let's see here. So let's say this is the intensity increasing, which will lead to increase in potential, receptor potential. As you can see here, the moment the receptor potential reaches the threshold, it will cause, yes, that's all or none, action potential. And the more intensity increases, oh, more action potential. The receptor potential increases gradually with the intensity action potential. As you can see, and you remember from what I explained in rapid coding, the more intense stimuli, it means more frequent action potential. So in other words, stronger stimuli, higher receptor potential, and the higher receptor potential, more frequent action potentials, which is, as you can see here, are the spikes. So how receptor potential are generated, how it happens in different receptors. Let's start with mechanical receptor. So as you remember, let's take one of the mechanical receptor, which is this receptor. Okay, so what happens, it's like, I will show you, it's like capsulated, Pacinian corpuscle. So it's capsulated. So what happened when you apply pressure or touch or just mechanical force, stimuli, it will cause distortion. These capsules are flexible and it has fluid in between. And this stimuli will cause distortion, like the movement in these fluids. And this movement will lead to opening of sodium channels. And with the opening of sodium channels, it will lead to generation of potential on the membrane. And that's what we call receptor potential. And then this receptor potential start to move locally. That's why it's considered local potential. And if you remember the difference between local potential and action potential from previous seminars. So it starts to move uh, towards the axon of the neuron. And until it reaches the axon and start to jump between nodes of Ramver, that's what we call action potential. So another difference between receptor potential and action potential, receptor potential are a local, action potential, it's moving. Uh, other, what about other types of receptor? For example, chemical receptors, from the name. So in chemical receptors, some chemical attached to receptor, which will lead to open of ion channel, or in other words, maybe start a second messenger, and this second messenger will lead to open of calcium channel and causing action potential. So the idea is just the different receptors has different gated channel, let's say ligand gated, voltage gated, and in this case it was in mechanical, it was the stretch of the cell membrane. So I want to imagine with me, that's a cell membrane. So when we cause mechanical stimuli, this mechanical stimuli will lead to stretch, and this stretch will open some ion channel, sodium channel, and this sodium channel will lead to sodium influx, and which will cause potential, increase in receptor potential. Other ways, for example, in test cells, which is chemical, I have test receptor, G protein, coupled G protein, which will lead to phosphorylation, opening of calcium channels, and calcium channels will lead to uh, the release of neurotransmitter, which will lead to action potential on the post-membrane neuron. Another way, which is a bit complicated, but I will explain in future questions, are in rods and cones, in photoreceptors, which we have rhodopsin. The light will cause chemical reaction of rhodopsin, and this change in rhodopsin will be coupled by G protein, leading to open of sodium channels, action potential, and so on. So that's about receptor potential and its relationship with action potential. So the stronger the receptor potential, the frequent the action potential. And remember, receptor potential are gradual, while action potential are following the rule of all or none. And that's why we can differentiate between intensity of stimuli. Uh, okay, that's so there is a concept of receptor and generator potentials. Some teachers say that these two concepts are the same. Others say they have some slight difference, but in the end, they are very similar. Both of them are graded potentials. Both of them are local, and they when they reach threshold, they will lead to generation of action potential. The difference is where they happen, their location. So receptor potential are happening in specialized sensory receptor cells, as for example photoreceptor and hair cells. If you remember from previous question, when I explained the primary and secondary, so usually receptor potential you can find it on the secondary receptor, and you can see generating potential in the neuro after the postsynaptic, on the postsynaptic membrane you can see generating potential, and lastly you see action potential. So let's say they gave you an exam, this picture, and told you, okay, point for me the three potentials. You will you will say receptor potential, it's happening locally near the axon. We have generating potential, and on the axon we have action potential. Here, receptor potential on the test cell, generating potential near the axon, and on the axons we have action potential. Okay, okay. So next is adaptation of receptors. What does adaptation mean? Adaptation is decrease in receptor cells response for constant uh, stimuli. What does it mean? So let's say, for example, you are going outside to hang out and you put some perfume. Okay, the moment you put the perfume, you can smell it very well, nice. But after some time, the smell disappears. But in reality, if you went and met your friends will be able to smell your smell, but you cannot smell it. That's what we call adaptation. Your olfactory cells get adapted to this smell. So because the level of this smell is constant or even decreasing, so at some with the time you get less responses. Why we get less responses? Because the number of action potentials start to decrease. Why they start to decrease? So that's why the adaptation has different mechanism depending on the receptor. Let's take Pacinian corpuscle as an example. So it has two routes of adaptation. First, when we apply mechanical stimuli, it causes distortion of fluids, as we said. But after seconds, these fluids get redistributed. So even if you have the force applied to the Pacinian corpuscle, now the fluids are returning to normal. So your stimuli is no longer generating potential, action potential. Another mechanism in this kind of mechanical receptor is that when you apply force, mechanical stimuli, these flexible membranes get adapt, and with time the sodium channels close by progressively close and get inactivated, which will lead to what we call adaptation. which is decrease in action potential. Other receptors, for example, in olfactory, so your receptors get the ligand gated channels get saturated with the odor odor molecules, the smell, okay, the smell molecules, which they just stuck on the receptor. They activate it for once, but after some time, the sodium influx start to decrease or even stop, which will lead to decrease in action potential. But some kind of receptor, some types of receptor, they never reach the zero adaptation. Like muscle spindle, joint capsule receptor, they have decrease in their response, but they never reach zero. Why? That's related to their function. Like for example, muscle spindle for their stretch reflex and so on. So let's revise again quickly the mechanism of receptor excitation. So we know that the different receptor has different mechanism. Mechanical, stretch of membrane, which will lead to open of ions channel. Chemical, lead to chemical ligand gated channel, which will lead to open of this ion channel and generation of action potential. Temperature. So temperature, what happened in temperature receptor is the difference change in temperature will lead to alters the permeability of membrane. What does it mean? So let's imagine before for heat receptor, not cold. For example, the temperature suddenly increased. Your body start to feel this difference in temperature that that the weather is getting hot. And this temperature will lead to that the membrane start to easily let the ions go in, especially sodium ions. And sodium ions, more sodium ions going in will lead to, yes, generation of action potential more. Because in temperature receptor, we know that they are not like, there is no receptor action potential and suddenly there is action potential. No, there is always action potential, but the moment there is a change, this action potential could increase or decrease depending on the temperature. And for light, it's a bit complicated, but I will explain it for you in the future questions. How light can break down rhodopsin, and this will start a cycle of ions movement in the receptor. And thank you so much for listening to me.